So good morning. It's still morning-ish. I know that I'm the person between you and your lunch, but bear with me just a little bit more. I have to start by thanking Walter and all the local organizers, because this technically he did this 3 times, right? So he did it for 2020, he did for the webinars, and then he did it back in 2023. So his patience was actually, and I know that he had a lot of patience to wait, so it was really cool. And now we have the chance to have this meeting again. It was really cool. I always remember when we started these meetings, especially in Telluride, but we always did these meetings with a sentence from Bertrand and from Marilyn saying that a very successful presentation is the one that doesn't pass the second slide. that's the metrics here. And I always say that sounds so nice. So if you have a question, you can interrupt at any point. I don't mind. Just raise a hand and raise the question. I will repeat it here, so we don't need the microphone. And because I knew that you might do that, so I will start my presentation with a bunch of slides that are just overview. You will not have the temptation to stop me at my second slide, right? So the whole point that I decided, I had an update on what I did on a webinar, and that is the subject I brought. I had a few other subjects, but my students, they stole all of them, so they will do their own presentation, their own stuff. So I'll have to talk about a similar subject from the 2021 webinar. And this presentation will focus, probably it will be the first one that will focus on constant pH molecular dynamics. There will be other people also talking about these techniques. So my point, I will not spend so much time talking about methodology, but for those that don't remember so well what is the stochastic titration, constant pH molecular dynamics. So this is a way for us to bring together Poisson-Boltzmann, Monte Carlo, and molecular dynamics. So if we bring them together, we can couple conformation and protonation. It's not always for all constant pH MD methods work this way, but in our specific case, protonation samples very fast, which is very cool. The slow part is always MD. So whenever we have a barrier, it's kind of MD takes a long time to sample everything that we want, and protonation tags along. So it's in some methods, I know that protonation might have the problem. With this method in specific, we have that advantage. Of course, this all started with Antonio. And it's a good thing that he just arrived, so it's there. he can hear. So the stochastic titration method is actually something that came out of his head. And when I joined his lab, he already had the method implemented in Gromos with this archaic Gromos 43A1 force field, but it was the best there was in 2002. And so my job was actually to bring it to the new millennium. let's say that way. So we had to go it from Gromos to Gromax. Gromos back then was, well, it's still slow, but it's not as slow as it was in the first decade of the 21st century. And our idea was to bring this to Gromax, and the new force field just appeared. The 53A6 had just appeared from Gromos. We were like, Antonio was a Gromos group, so I only learned about Charma number much later. So we just did everything with Gromos, and there was a few tricks that we implemented. We wanted to make it fast. Gromax was incredibly fast back then, comparing, and all of these developments were happening in Antonio's lab, and we decided to include tautomeric because we realized that we could protonation sample very fast. The movement of a side chain of an amino acid could take much longer than the frequency at which we were trying to stick a proton to amino acid, right? So we just decided maybe we can speed things up if we just introduce tautomers. And tautomers is not only for histidines, they were just for carboxylic acids. So a carboxylic acid in our method has always 4 positions where you can stick the proton. We can sample all of them at any point. So that was also something very, very interesting. It improved sampling. And 2011, this was like a major wrap up of things that we did together. And here the major part, of course, there was the new force field coming up from Gromos, which is still the one that we are using, 5047. But the interesting thing was we decided to calibrate, because until this point, we were still using PKs from analogs of amino acids from Tanford, you know the numbers. And we always had the feeling that it's not really the best thing, the best approach. And at this point, we decided, okay, let's do some tricks. We can use these pentapeptides from Nick Pace's lab. We have the PKAs. We can run the pentapeptides constant pH MD. We do the calibration. We say so. So for the pentapeptides, we are on the mush, and on the protein, let's see. And that improves quite a lot our predictions. We are always 0.7, 0.8 pK units RMSE in almost all predictions, and that's like best case scenario. And that's, you have to live with it. And of course, when most of you met me in Telluride, and I was the guy that was always making fun about the name of the meeting, protein, electrostatics. I always have the first slide saying proteins, and then I include, and lipids, electrostatics. And the reason is I was titrating lipids back then, and for that, I needed a PV solver that was much faster than Mead. So I migrate into Delphi. And Delphi had this ability to include periodic boundary conditions on the potential calculation, which was also very good. And I spent a few years doing titration of lipids. Titrating lipids is really cool. It's very important for many processes. But what we really were interested is to know if when you have molecules interact with lipid bilayers, what is happening with the proton behavior of those molecules, but also what is happening on the protonation of lipids. And for phenomena like, let's see if it changes the slide. No, it didn't change the slide. It means that, okay. So what's going on here is that when you have a peptide, in this case, we're just the simple pentapeptides that we use for calibration. But here we are just interested in studying the interaction of these pentapeptides in the membrane. And most of these peptides, they do not only absorb, they tend to really insert and go below the phosphate level in the membrane. And we did a lot of calculations, and we substituted, it's like a pentapeptide of alanines. We just substituted alanine in the middle by a titrable amino acid. And what we wanted to know is, how does the pKa change? Or like some other people like to say, how does the pH change? Which is another way, it's like it goes around, right? So when people tell you that pH is lower near a membrane or an ionic membrane, you can always say the pH is a bulk property. It's what's changing is the pKa. So if you just clean up that out of the way, you have to think about or you can think about as pKa's, as energy proton binding affinities. And it's cool that we could really get an idea of how much does the pKa change when these amino acids insert into the membrane. So this was cool stuff that we did with very poor sampling. We've been always trying to improve the sampling to get much complete, because we know these pentapeptides, they go much deeper than these values, but we could only calculate pKa's when we have enough ionized species, right? If you don't have a population of ionized, you cannot calculate pKa's. You always have neutral form. In neutral form, they can go much deeper than -6. They can go to -12, -10. Then it's a wall. It's a dry wall. And the thing was, back then I was just moving into these more interesting systems where pH was important, because going from pentapeptides interacting with the membrane, we always wanted to study membrane proteins, right? It's always been our aim. We want to study membrane proteins. So the first thing is to start with like peptides that are transmembrane. And in this case, it was a flip peptide, which is really cool because it's a peptide that when you protonate a specific amino acid located here, if you protonate it, then you stabilize the helix. If you deprotonate it, if you increase pH, then it will ionize, and the whole thing starts to unfold and exiting the membrane. So it's a membrane inserting peptide that is activated by acidity. And it's very important because of tumors, right? Tumors, especially very advanced tumors, they tend to be acidic. They create this acidic environment. So these peptides have been used to labeling and including for some therapies, but mostly for labeling for us to see the tumors. And in our simulations, we did something similar to what we did with the pentapeptide. So we could actually follow, and this is not so obvious, but it's once I explain, maybe it becomes obvious. If you go inside the membrane, you can follow the pKa, and the pKa goes from 4, right? It's 3.9 to, in this case, to around 6. Well, we knew that the peptide followed by fluorescence, we knew that it has this transition. It inserts into the membrane once the pH comes lower than 6. So we already had a value, and it's very simple. If deeper inside here, think about this aspartate. When the aspartate is inserted into the membrane, it does not see water. If it does not see water, it does not see pH, right? So if it does not see pH, there's no membrane. It's neutral. So the peptide is happy. So when the aspartate sees water, sees the strength of the chemical potential, so it sees the pH, sees the amount of protons. If it loses the proton, then it promotes an exiting. So it's always an equilibrium, and this is how we interpret this data. So we look at the most deepest that we can calculate the pKa, and then we just assume that this is the triggering pKa. Of course, if this aspartate is in this position, the pKa will be different, and it will not correlate with any experimental data because there's a moment of triggering, and that's when the head is outside of the water, right? In this case, it's the wrong analogy. It's the opposite because the water is on the top part. So we start having problems with sampling. So what we did was we just started to couple constant pH molecular dynamics with enhanced sampling techniques. And this was a cool one. So we use replica exchange. And with replica exchange, we could improve significantly the convergence of our simulations. We could actually get away with only one replicate almost. Running just a single replicate, we could with simple constant pH molecular dynamics, it would be almost impossible because you start having problems where protonation decreases when you lower pH, which is something strange. And it has to do with because conformations of lipids, they take a long time, and doing simulations of constant pH MD simulations, single simulation becomes problematic. And in proteins, it converges much faster. Once you go into confined environments, then you start having problems. And then Pedro came along. He's over there. Pedro was a former PhD student. He's now working in bio. So he went to the dark side, to the machine learning world. And Pedro developed something very cool. So because we had all this methodology around Delphi, we had built a thermodynamic cycle for Delphi. We could calculate easily pKa values. So Pedro did that easily with a Python tool. We also had the idea of doing the server. So every time you use prop pKa, I should like hit you. Try to use Poisson-Boltzmann. It's almost as fast, almost as. And this is Poisson-Boltzmann, so you should. It always gives better results than it's not instant, but it's almost instant. If you stick a PDB code into this system, it was already pre-calculated because we calculated a big portion of the PDB data bank. It's already in database, so if you just stick there, it will give you it's like doing the running prop pKa, right? And we did this server. From the server, we did the stuff that I was telling you about. We built the database. The database is also accessible. A lot of groups are downloading the whole database so that they can train their AI models and doing a lot of stuff with it. This, of course, once we had this, we also decided maybe we should also make an AI model for this. And this is what Pedro did. Pedro built a simple model. I think if you have any deep questions about this, I think we can ask to Pedro, because he's kind of on holidays, but he can answer some questions about the project. And the thing about this, the thing that most puzzles me is most AI models, especially for PK predictions, are based on the experimental PK data available, right? But there's around 1,000 numbers. So you can train, you can overfit, you can do whatever you look, or try not to overfit, which is a challenge with those 1,000. And the worst part is that those 1,000 numbers, which are pKa values that are in the experimental database, they are mostly correlated. They are almost all from a very short, a small number of proteins. So it's like bad on bad. But that does not tell you that you should not try. Everybody should try and build a model out of it. And the model works reasonably well, at least from all the papers that I've seen. But we did here something different. So we want an AI model that can predict the Poisson-Boltzmann estimative. You see, it's a different thing. And once we did that, then we have an AI model that can predict the PBMC values quite nicely. And then we decided, so what do we need to do to get, from this point, to get a good estimation of experimental PK values? The factor is a dielectric constant. What we did is attenuate the bad guys. There's always bad guys. So we just need to introduce a regulation factor, and we can get, out of it, we can get pretty decent PK estimatives. So that made us think that maybe there's some merit in, if we have physics-based methods that can do good predictions, and maybe then we can generate enough data to train AI. And the advantage is we can calculate the pKa values of the complete PDB data bank in, well, maybe hours. I don't know, but not that different. And this was always the goal. Is this slide? The whole goal was to have our one pKa predictor that could be coupled to molecular dynamics and create a constant pH molecular dynamics that is easy to use. If you know how to use GROMACS, you know how to use this, because everything is wrapped up, everything is optimized. And this is our future constant pH molecular dynamics method. So if you know how to, the input file is just one, and it's an MDP file. So if you use GROMACS, you've seen an MDP file. It's an MD parameters file from GROMACS. So it's that it has some, of course, it has to have some extra settings, right? At least you need to say which pH you want to do the simulation. So that's an extra, but it's very easy to parameterize and to use. And of course, we use replica exchange. We also couple it with umbrella sampling. And with umbrella sampling, this is the work of Nuno. And he will talk to you about this in the afternoon, so I will not take much. But the whole idea is there are some phenomena that you are limited by the MD. So under restrained MD, nothing of these like transport processes will work. So we need to couple them, and we can couple them with constant pH molecular dynamics, which might answer a few of the questions and some of the things that were discussed already this morning regarding this. We can do this. We can not only follow the PMF, but the PMF will be calculated at a specific pH. And then you can change pH, protonations, and the microstates will change. And then the PMF will change also. You see, we can get all of this. And these are just examples. This is only at one pH, but it has the difference between running ADP on one direction, ADP on the other, and then ATP on the two directions. And we could actually see difference at a preference for ADP when it's introducing inside mitochondria, and on the opposite, it's not needed because there's a lot of ATP that needs to be, because ATP out of the mitochondria, it's easy because it's in favor of a gradient of concentration. ADP from the cell into mitochondria, then it's much more difficult, and then that's where the selectivity comes along. And another thing, this is just a trick. This was me borrowing an idea from Antonio, who also borrowed probably from Ullman. So pH gradient, it's very simple. You just define two groups of sites. Some sites are titrating on one pH, the other sites are titrating on the other pH value. And by doing so, you can actually have a gradient. pH. So we can couple that with constant pH molecular dynamics and run everything at the same time. And this is also cool. And we did this for the same peptide I mentioned, flip peptide. We did this where we kept the pH on the inside of liposome. This was still for liposomes. On this side was one value, on that side was another, and you could see that it makes a difference. It actually makes a difference on the predictions, and it 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 helps understand the difference between what's happening in cells and liposomes, because we know that this peptide works very nicely in liposomes, but when it goes into cells, they are not being used. There are different sequences that are much better than the wild type sequence. And the reason probably is one of the many factors, like lipid composition, and one of the factors can be the pH gradient, and we showed that. And these are just 2 or 3 slides just to show the people that are going to talk. And in the case of João and Flip, they did the extension to the charm force field. So now we can do these constant pH molecular dynamic simulations using charm 36M, which was a major improvement until we actually use it, and then we realized charm as a much more sticky than Gromos. Gromos is a blessing because it's much more fluid, it's united atom, but charm, it's more sticky, so things, when they stick together for longer times. So it's just that it's not a problem of charm, it's a problem of compatibility with our approach to constant pH molecular dynamics. So things, I give this example is because both force fields start with the wrong prediction because the X-ray structure was not correct. And then within 50 nanoseconds, Gromos corrected the pKa of the cysteine with the charm. I'm sure that it will eventually lowers the pKa, but it stayed for 50 nanoseconds without nothing happened. It's hydrogen bonding and it's very happy. Yeah, but you have the pKa's when you reduce, you just put the residuation. Yeah, and you have 2 structures. You have a structure where it's oxidized, another one is already reduced, and cysteines are slightly apart. Here we use on purpose the structure from the oxidized, so they are 2 together. You see, so you have to force it during constant pH molecular dynamics. You have to force it to come apart so that they decided to ionize because they don't. Yeah, but you know you can break a covalent bond in the computer. Yeah. So and this is what we are doing now. So now we are extending to Amber in collaboration with Adrian. I tried to convince him to come in here more than once, and you too. Yeah. And he was supposed to. I think he's in Europe right now, but he didn't have the chance. But we are now doing this. This is the work of João, who is also over there, and he will talk about this in detail and what he's been doing. And this is Nuno's work with the Reus. This is our idea. Okay, if you have replicate chains, if you have umbrella sampling, we can bring them together and couple them. So this is just hybrid methods that we are trying. We always need something extra, and this is the things extra that we have been developing right now. And we've been applying constant pH to everything. And I think I am too good of a boss, because they always bring new systems. I should say no, but I never say no. So they have very curious. So this guy wants to continue some old work I did with Antonio with dendrimers. So Flip, the crazy guy with dendrimers, but he studies the effect of pH on ramified molecules. There's no secondary structure, but there's a lot of there's a lot of coupling with the protonation. Sarah is studying on protein knots, which is a completely different topic. You can learn more about it. And João is also studying protein aggregation, so interaction, what is stabilizing 2 monomers bringing together forming dimers. And here are just the guys with the posters if you'd like to look at it. There's tools, there's constant pH molecular dynamics, there's drug binding to a receptor, and of course one of the most nicest posters. The other ones they don't you will not take they will not take a bad opinion if I say this, but this poster I think it will intrigue you. So what Ines is doing is something that I think all of us already thought about, if you pick up a PDB structure and you do prediction, and then if you pick up a second structure and do a prediction, how much will that affect? So what, because we had a tool to calculate pKa's, so what Inês did was just run over a series of proteins, all the PDBs that are available, and check out how much does pKa prediction vary in that ensemble, and at the same time say, okay, and what if we want to compare with experiments? How well do all of these structures compare with experiments? Are there a better structure or a worse structure? And there are. You can go and check the poster. And now you can start to interrupt. So this is just an update on the previous work. What we have here now is a study that we've been doing in the lab, trying to model how does the acidity impact the behavior of molecules when they interact with the membrane. So it's a continuation of most of the things that I already said here. And I think I already made a case. So when there's tumors, usually there's a slight acidity. It's a very small acidity, right? So we are talking about 7.4 to 6.2 if we are benevolent, but it's a very small difference. So let's call it 1 pH unit difference. But this 1 pH unit can generate, yep.
Sorry, just to be clear, you're talking about cytosol or extracellular matrix?
So in this case, this is the outside of the cell. Okay. Yeah. Yeah, it's a difference to the outside. So it's the tumor cells, the cancer cells, they accumulate in these small tumors, and then because of that's very bad perfusion, so blood cannot go there, and they accumulate because yeah. Yeah, it goes from 7.2 to 7.5 in some yeah. It's very small, but people say that the going up inside the cell can actually be more drastic or dramatic for cancer than actually the outside. But we want to study the impact that these have on therapeutics, right? So if we consider this slide, it kind of sums up things, right? You can see the 7.2 to 7.5 on the inside. On the outside, it acidifies. It can acidify a lot and less. And we know something that is a very strong hint is the more acidic it becomes outside, the poorer becomes the prognosis of the cancer. So there's a correlation done maybe 15 years ago, and so we know that. So the more acidic cells become, more difficult it becomes. They become more resistant, and they become more aggressive. All of those characteristics, and so we know this. We know that there are lysosomes that have a low pH, and because in cancer, what is known that lysosomes do is they usually trap a lot of, it's the garbage can, right, of the cell. So they trap everything that we throw. If you throw at cancer a lot of drugs, they tend to come and end up here. And what cancers do is that when creating resistance, they just create more of this. So in cancer cells, there are much more lysosomes than in normal cells. It's one of mechanisms. Of course, they overexpress exporters that are always kicking the drugs outside the cell, which is probably number one. For most people, it's number one reason why there is resistance to drug therapy. These are just 4 examples, very typical drugs. I chose them just because they are Lewis bases, okay? And if you think about it, all of these have this amino group that have a pKa between 7.5 and 9.5 or 9, and they are all being used in cancer therapies. And if we concentrate on, for instance, sunitinib, has a pKa of 8.5, and I don't need to explain what this means, right? So it means that at pH around pH 7, it will be 95% protonate. And this is very cool because it's the only way you can give this as a drug, even if it's injectable, because you need it to be soluble in water, right? So that's why Lewis bases are so tempting, because they become protonated, ionized in water, very soluble. Because usually the other part of the molecule is very hydrophobic, is not so soluble. So when these compounds are going to get into the cell, of course they need to become neutral, because otherwise the energy barrier probably it's too high. I never measure it, but I just assume that it's too high. And it's not so complicated. It's a Lewis base. It needs a small shift in the pKa, loses the proton, and then it can regain the proton on the other side and actually helps getting away from the membrane, because remember, they are very hydrophobic. They probably would like to stay a lot in the membrane. So the fact that they can protonate and that will gives them the push to get away, it might be an advantage. So everything is perfect. If you think about it, it's perfect. In MD simulations, we cannot simulate the crossing because it's too large. The barrier is too high for MD. Probably kinetics, it's much slower than nanoseconds. That's what we do. What we can do is you can use the constant pH molecular dynamics and calculate the fraction of protonation, means the fraction of charge. So when they are in water, they are like half charge at pH 8, but at pH 7.5, you see that it's like 90% protonated. But once it's in contact with membrane, remember dissolvation, neutral form becomes more stabilized, and then it goes really deep in our simulations, but always has a neutral form. So it makes sense. The whole mechanism of deprotonation crossing the membrane might make sense. And you can go to pH as low as, like in this case, 5.5. And what it says is it starts having a problem. If you look at the abundances, which are spotted here, what's happening is that at lower pH, you start having problems sampling the most deeper regions. Makes sense. It's more time protonated. It doesn't go so much. It doesn't like so much to go too deep into the membrane. So the abundances are kind of two sweet spots, one for the neutral form, the other one for the charged form. Because we have all of this pH, we can calculate pKa values. We could calculate for several. So we see a shift of the pKa. So it's the energy. How much does it like to have the proton? And if you think about the 3 pH values we were talking about, normal cells, tumor cells, lysosomes, you see that if this is a metric of energy, you see that it becomes, the lower the pH, it becomes more difficult to deprotonate. And so that makes sense why these drugs, when they go into lysosomes, which is here, they become trapped. And they cannot get away, because then it's much more difficult. To go in is easy, to get out is difficult. Think of it as a sink that will attract them. For us to study the whole process, we needed to couple this with umbrella sampling constant pH molecular dynamics. And by doing so, we actually can have the complete profile. We can calculate the PMF. And these are the PMFs at different pH values, which are really cool. What can we do with these PMFs? We can calculate membrane permeability. We can actually go into this diffusion formalism and get the permeability coefficients. And permeability coefficients, even though the difference in the PMF are small, they are incredibly different. So that's an order of magnitude between a normal cell and tumor cell. So what this tells us is that when these molecules are encountering a tumor cell, they have 10 times, in terms of, let's say, probability, they have 10 times more probability of going into a normal cell than they go into a tumor cell. So we are probably using these compounds to treat cancer, and we are using concentrations that are at least 10 times higher than we need to, and generating a lot of side effects. But side effects, you all know about them, right? So, and what is the consequence of this? This is just an explanation of how the protons are concentrating. So these are the exported, but this is the slide. This is the target for sunitinib, so right? So one of the targets. So KDR gene gives up this receptor, and this is the spot. This is electrostatic potential mapped on the surface. If you look at it, you say, there's no reason why it needs to be protonated here. Where is the advantage? There is no negative electrostatic potential at the entrance of the target enzyme. So it was introduced here because someone, maybe 20 or 30 years ago, decided that they needed a Lewis base there to increase solubility. Yeah. Okay. And the whole point of this slide was to show you that we could actually think about, instead of using a Lewis base, trying to use a Lewis acid. Probably carboxylic acid is not the best example, but if we use a Lewis acid here, there is a good chance that we could accommodate that compound in this pocket, And instead of like the amino group is looking for the negative, the acid that is probably located on this position here. And instead of looking for this, we could probably see what's happening is that amino groups located on this position are actually looking for the negative charge over there. So this means that the pocket can accommodate to whichever charge that you have there. So in terms of electrostatics, it would make no difference. What is the main difference in terms of permeability? If you change an amino group for an acid group, you invert completely the selectivity. So that, in principle, we would have a compound that has 10 times more permeability higher than at pH 6 than it has at pH 7. So the permeability would increase when you lower pH. If I make myself, and this was just a docking slide where we actually compare to see if docking could actually see the difference between the amino group and the acid group. And there aren't many differences, and it's docking, right? So even just to change protonation state, if we decided to do that, it would be a problem. And this was my last slide. And let me just thank all the guys. So most of the people that are in this slide, they are here, so you can you can say hi and talk to them. And let me thank the funding and you all for listening. Thank you.
Thank you. We have time for a couple of questions.
Hi. Oh, I wanted to ask you why you didn't choose a mixed lipid bilayer with some anionic lipid, like with POPC and some anionic one.
It's a good question, especially because I know how to study the effect of pH on those lipids, the anionic lipids. I did that. The answer is because I already did those mixtures, and I know what are the limitations of force field. The moment we start to introduce anionic lipids, and cholesterol would be a no-brainer because we will need to study the effect, and parameterization of cholesterol are really cool. But the thing is, if you introduce anionic lipids like POPS, if you start introducing, if it's a concentration that is low enough so that the force field effects do not appear, the impact is not as high. If you go into more after like 10% of PS, you're starting to have effects on the electrostatic potential. PS starts the charge of PS starts to deviate. It's no longer the charge that we expect to be -1 altogether, and you start to see those effects. And then we would need to simulate the lipids with constant pH molecular dynamics, allow them to change protonation states to have it more realistic, and then rely on force field parameters. It's a trade-off. So it will have an effect. Most of these pKa curves will be shifted. Altogether, they might not change much in shape, but it will be shifted. The acidics will be more that way. The basics will be more on this side. Yeah, we plan on doing that, but we didn't do it yet.
Okay, thank you.
Great talk. So about a small comment about the sticky protons in charm 36. So in NAMD, when we were still developing constant pH, one problem we found was it's exactly the same problem. Things were sticking. It was sticking less for soluble systems. As we moved to transmembrane systems, there was hardly any exchange. It was sticking a lot more. So we started using something called the Galvani potential in order to, as a correction. Of course, in NAMD, our methodology for constant pH is a little bit different. It's not lambda dynamics. It's non-equilibrium switching. And then with the usage of that minus 520 electron volts in the correction factor, things started moving a lot better. I don't know if it's a charm issue or specifically a difference in methodology, but that term literally helped us.
Yeah, so I saw the paper, so I know. What I can tell you is, I know that things improved with your approach. My argument is that the standard constant pH molecular dynamics with charm should work. Yeah, I think it actually should work. I still don't know the reason, and you know that implementation of constant pH molecular dynamics in GROMACS, that implementation is focused mainly on charm. Yeah, and
so I can comment on the Galvani potential. Galvani potential is a correction that's made to compensate what Tom talked about. Maybe I can also discuss a little bit tomorrow from a different angle, Galvani potential angle. It has nothing to do with what he's talking about. A. B is that you only need that if you run PME. So all item PME constant pH, and he's doing a hybrid approach where his electrostatics is calculated using Poisson-Boltzmann.
We use PME.
Yeah, use PME, but he's using Poisson-Boltzmann. So the PME part is not really affecting because you don't have explicit water to give rise to the.
No, we don't. But the fact that
potential,
the fact that a nitrogen bond is very long-lived in charm.
This has nothing to do with Galvani potential.
Yeah, well,
there's a shift about the unit. So depending on the box size, but we tried out. Maybe I can talk about tomorrow various box sizes, but works out to be about 1 unit for all residues in one direction.
You're correct. It has nothing to do with but the question was, how does the galvanic correction approach improves the sampling? And in the paper, they showed that it improves significantly the sampling.
It's JCP paper, right?
Yeah, that's the
that's not sampling.
with
and there's something wrong, honestly. I communicated with him. I calculated his box size and I came to a different conclusion. So I think there's calculation or something not quite right.
I see. We can talk about that.
I talked to Benoit about this.
Yeah, the thing was before using the Galvani potential for transmembrane systems, it was not, there was no exchange at all.
Yeah, so whoever did the calculation overestimated the correction by 2 or 3 units.
And the basic difference is, of course, lambda dynamics versus this non-equilibrium.
Again, this has nothing to do with
okay,
this
fine.
potential. Yes, it's exactly what Tom talked about, the model system versus the protein system where you have less water compared to the protein volume. So the hole is very big in the protein system. Whereas in the model system, the hole is very little. So you can consider the entire box as if it's water.
I see. Thanks.
Okay.
Well, actually very interesting. So you provoked a lot of discussion now. Actually, I was thinking also between the 2 talks and the boundary conditions for the. Basically, if you do a standard Poisson-Boltzmann calculation, your boundary conditions on any box, any size we're using would be Coulomb, right? You will never set 0. And clamping down potential in the water simulation is something which can be easily corrected for. Now, the question for the membrane simulation, I would think, as much as I know about membranes, that all the problems of boundary conditions on the electrostatic potential on the boundaries in the presence of the membrane will be hugely exaggerated. That is, they will increase rather than decrease because there is much less shielding. So all the electrostatic interactions in the box, which also has membrane, are longer range than in water.
Yeah.
So then when you mentioned the lipid, actually, that's where I started thinking the charged lipid probably is also problematic for that reason alone.
There's only one way to do it correctly, which is to introduce periodic boundary conditions on the potential calculations.
Right.
That's what we do.
In the simulations.
Yeah. So
but not PME, because PME will clamp it down.
Yeah,
right?
That's true. But,
but then that's my question. Then.
We have those simulations then with reaction field, with PME, they pretty much give the same results.
Have anyone tried reaction field, like go back and do reaction field in this field?
We do it.
And I would think it should work better. Does it? No?
Yen already changed her mind regarding reaction field.
No, because basically that used to be fixed, right? You won't get a correct dielectric constant of water unless you set up correct reaction field. Or do Ewald, which is actually a fix, just like
membrane
your
is
galvanic
debatable,
potential.
and,
Ewald is just a
we
fix.
all agree that it's not the best approach to do it in membrane, but
yeah.
But it actually works. Yeah, it works.
Mhm. That's just that the actually popularity in the community forces some standardized errors in the field. Like Ewald does problems, and it became kind of you have to do it, otherwise you're not. Well, it depends. Yeah. Yeah, enforced. Okay.
Real Telluride discussion here going on, and people waiting to go for lunch.
Okay.
No, I don't have a good implementation, but if you could, it would be interesting. It's just a question. Right.
Okay. I think we are getting hungry.
It's the understatement. I can confirm there are no questions in the chat.
Okay, thank you.
It's a quick.
So have you talked to Karen Fleming about this, your work with the peptides and PKs in the membranes?
No.
Oh, okay.
Karen Fleming?
Mm-hmm.
Don't even know the name, I'm sorry. Probably bad of me. Okay. I might have seen the work, but I cannot relate the name. Yeah, we should.
Okay. Thanks again, our last speaker of the morning.